The paper I have selected for today's review is based on the establishment of a suite of benchmarking tasks for protein sequence fitness prediction. These benchmarks are known as FLIP, that is, Fitness Landscape Inference for Proteins. Before delving into the work of authors, I would like to focus upon the need for this particular benchmark. Whenever a new generative or machine learning model is trained, it needs to be evaluated upon its capabilities. This evaluation requires some benchmarks against which the model needs to be tested. Herein, FLIP enters the picture with two of the major criteria, that is mutations that are beyond a parent sequence or that lies in a complex landscape that involves multiple amino acid insertions and deletions, while also accounting for the functional diversity of the sequence space. The authors worked with three groups of baseline algorithms, that is parameter free, supervised, and pre-trained models to generate a set of benchmarks for knowing if a model covers both sequence and functional diversity. One of which is partitioning the landscape or splitting the entire dataset into different train and test splits. The authors considered three different landscapes and had different motivations behind each of them. They divided the entire data into 15 train test splits on the basis of biologically relevant generalizations, such as the splits could mimic the challenges seen in protein engineering, like the thermostability landscape had splits over clustered sequences of mixed species, human species, and a particular cell of humans. The three landscape begins with GB1, a binding domain of an IgG molecule, where they looked upon the effect of epistasis, that is, effect of interactions between the mutations. The focus here was on learning variants with fewer mutations to predict the properties of variants with more mutations. With adeno-associated virus, that are capsid proteins responsible for virus integration into a DNA payload, they look to predict mutations only at subset of positions, like at a protein-protein interface. With thermostability, the main aim remains to study both global and local variation, as even a single amino acid substitution can confirm or destroy the thermostability. By testing these splits with different baseline algorithms, the authors concluded that randomly splits dataset oversplits the results by a huge margin, and that random splits reflect a notion of generalization that is not of interest to the biologist, while a more accurate way would be to keep the biological mimicry in mind while making the splits between data sets. And they also identified that pre-training approaches perform well on tasks with diverse sequences, such as thermostability, but they do not outperform simpler supervised models